So, depending on where you are, I wish you a great morning or an excellent afternoon. It's my great pleasure to welcome today Dr. Soumen Basak as another excellent speaker in our Global IMMUNOTALK meeting series. I would also like to express a special thanks to him because he joins us essentially at 9:30 p.m. his time and still gives his lecture live, which is great. I would like to say a few words about his career. So he started with a bachelor in chemistry, switched quickly to biochemistry, did a master and PhD at the University of Calcutta in India. At the time, Soumen studied mechanism regulating expression by Chandipura vesiculovirus, an emerging human pathogen in India. Afterwards, Soumen moved to the University of California in San Diego, where he joined the lab of Alexander Hofmann, and there he was very successful in investigating novel mechanisms that regulate NF-kappaB activity. And that was from 2003 to 2010. Then in 2010, he moved back to India and became a professor, a PI at the Systems Immunology Laboratory in New Delhi in India, where he is since then. He has been very successful in this position. He was awarded extensive funding from the Indian government and for projects, which I'm sure we will hear quite a bit today. He received numerous awards and is remarkably member of three different national societies in India. And if I would have to summarize his research achievements and activities in one sentence, then I can say that he has been very active in studying how complex immune signaling mechanisms, particularly those involved in NF-kappaB system, modulate inflammation in physiological environments and how dysregulated cell signaling pathways trigger abnormal inflammation in human diseases. And with this, it's again my pleasure to welcome you. And as most of you know, we start our lectures usually with a career-related question. So if you don't mind, I would like to ask you if you could share with us your most impactful decisions or decisions on your scientific career. Thank you.
Thank you, Professor Jain. And it's really a pleasure to be here. And I have to also thank Carla and Elena Zuniga for this wonderful opportunity to present our work. And yeah, I mean, starting from your PhD days and then a long postdoc and then a long, at least more than a decade-long career as an independent investigator. When you look back, I feel that one decision that may have made a striking impact, of course, the transition. I mean, I have been extremely benefited because of my association with our mentors, my PhD advisor and postdoc advisor. But the decision to join Professor Alex Hofmann at UCSD was one defining moment in a retrospect I have to acknowledge. At that time, he was new. He was just setting his laboratory. The day I joined, he assured a lot, but I think the day was a bit devastating. He had a picofuge in the entire lab in a building which is completely empty because it was a new building. It was not a lot of things going on there. And when you look back, ironically, that was actually so beneficial. PI had time and we discussed science, we discussed techniques, we discussed how to set up groups, we discussed how to address bigger questions or day-to-day routine laboratory activity, how one can manage research. And those first few months or maybe almost a year, that association continues to be a treasure for me. When I look back and I think, and I run my lab, I do my research, I design my experiment, and I think that was a bit interestingly came out in my favor. Although I joined a lab where initial first few months it couldn't be too productive because you had to set things up. But those associations, those interactions would be difficult for today if anyone joins his lab, he's director of institute in UCLA, won't have the time. And that I think, it was not that planned that way, but it happened that way. And I take pride in that. I was his first postdoc, and he is also, he acknowledges that fact. And, that association and that training would be otherwise difficult to achieve if you simply go by professional goals. So that's one thing I wanted to share, and acknowledging that I was, of course, extremely fortunate from my PhD advisor, Professor Dubojyoti Chattopadhyay, the way he mentored as a PhD mentor, PhD supervisor. Those were difficult days. You were learning things, were failing. Those are really important. But again, mentor association, particularly the decision to join a relatively new lab, which was extremely beneficial for me. I think you're muted.
Yes, sir. Thanks a lot for sharing those insights. And now we all look forward to hearing your talk.
Yes. So I will start sharing the slides. And please let me know that if you can see this well.
It looks all good.
All right. So yeah, I mean, it was not. Let me start this conversation here. I will probably try to articulate what we do, but I don't think it was planned this way. But today I am presenting, and today, just a few hours ago, something remarkable happened. India became the fourth country ever to land a spacecraft on the moon. And as you know, that's a moment for pride for Indian science and scientists, and of course for every Indians. And I just wanted to sort of share this excitement that we can perceive all around us in the institutes and in the neighborhood. Nevertheless, going back to what I wanted to talk about, cell signaling crosstalk, something that I will come in a minute, and particularly I'll be discussing the role of the non-canonical NF-kappaB pathway. So before I go into that, this is the institute, National Institute of Immunology, where I ran my laboratory. I've been here for around 13 years now. And this is, you can see, it's a beautiful campus in the middle of Delhi. And this is the laboratory that I head, Systems Immunology Laboratory. We fondly call it SIL family. And mostly driven by PhD students, enthusiastic, energetic, and very aspiring PhD students, graduate students. And this is my current group, and some of the alumni members, their photos you can also see here. And we study, even in our, of course, research, we collaborate with cohort of other groups in India and abroad. And without their help, it would have not been possible for us to achieve what we have achieved so far. And I also have to acknowledge funding support from Department of Biotechnology, Government of India, National Institute of Immunology, our institutional core support. Now, going forward, back to the science, inflammation must be tightly controlled. There's no question about it. You have microbial, just to refresh your memory, you have microbial ligands or inflammatory cytokines. They activate dynamically regulated intracellular signaling pathway, immune signaling pathways, and that instructs controlled expression of chemokines and cytokines. And that's the very essence of innate inflammatory response. This is very important to protect. This is very important to limit local infections and restore homeostasis. However, sometimes these pathways, their activity, you can find that their activity becomes deregulated, and this aggravated intracellular signaling would lead to inflammatory pathologies, for example, inflammatory bowel disease. On the other hand, if you have insufficient activity of these immune signaling pathways, it would compromise host effects. The question that we have been asking for in this laboratory is how this intricate control over inflammatory response is achieved. And in this, to this end, we, in our research, we have particularly considered this cellular signaling network. As such, one can imagine there is an instructive pathway that directs inflammatory gene responses. For example, S1 that I have been trying to, I have articulated here. But one can also imagine in cells in their anatomic needs, there are other ancillary pathways as well that are activated by tissue microenvironmental keys. And what we have gathered in last decade or so, 2 decades, that these pathways are not insulated, they are connected by a variety of physical and functional means. So in this laboratory, what we have been discussing, because these pathways are interconnected, if ancillary pathways thereby modify the dynamical activity of these transcription factors. If these ancillary pathways, because of their interconnectedness with the instructive inflammatory pathway, would they modify inflammatory gene expressions? In essence, what we have been asking is this: the central hypothesis that we have been testing for the past decade or so is this: if crosstalk between cell signaling pathways tune inflammation. And when it comes to immune signaling pathways, obviously we have focused on NF-kappaB signaling. Within the NF-kappaB system, there are two arms. The instructive pathway that plays an important role in inflammation is called the canonical relay NF-kappaB pathway. Part of this pathway, you have this latent complex, where the relay containing NF-kappaB heterodimers are sequestered by its inhibitor I-kappaB alpha protein in the cytoplasm in these latent complexes. Microbial cues or cytokines activate this kinase, NEMO-IKK kinase complex, that phosphorylates these inhibitors, liberating relay into the nucleus, where it activates dozens of inflammatory chemokine and cytokine genes within the canonical pathway. Relay NF-kappaB also induces synthesis of its own inhibitor I-kappaB alpha that generates a negative feedback that ensures appropriate post-induction attenuation of such inflammation-provoking canonical relay NF-kappaB activity. So, and one can easily measure, conveniently measure the activation of this pathway. For example, if you treat cells with TNF, that would lead to canonical NF-kappaB signaling, whereby I-kappaB proteins would be degraded. That can be measured by immunoblot analysis. And one can also harvest cells, prepare nuclear extract, and analyze them by electrophoretic mobility set assay to determine nuclear activation of this relay-containing dimer, as has been shown in this EMSA gel. So that's the instructive pathway. That's inflammation-provoking canonical relay NF-kappaB pathway. On the other hand, within the NF-kappaB system, there is also an ancillary pathway. We call it the non-canonical RELB NF-kappaB pathway. Its important role is in immune development, immune differentiation. Traditionally, it is not thought to be a major player for inflammation. At the heart of this pathway, you have this gene called NF-kappaB2 and this protein called PIONA-100. I'll be talking about them a lot. This PIONA-100 protein is also otherwise known as NF-kappaB precursor. This non-canonical pathway is activated by a subset of TNF receptor for superfamily, for example, lymphotoxin beta receptor or CD40, that would activate this kinase complex termed NEK-IKK1 kinase complex. That would phosphorylate PIONA-100. It would lead to, peculiarly, its partial proteolysis to a mature NF-kappaB subunit P52, which is known to bind to RELB, appear in the nucleus and alternate RELB NF-kappaB DNA binding activity. Unlike dynamical controlled canonical REL NF-kappaB activity, non-canonical pathway activated RELB activity, NF-kappaB activity, it shows a slow but sustained and very weak nuclear activity profile. And these RELB containing dimers are known to regulate expression of lymphokines, for example, CXCL12 and CXCL13, thereby they play a role in lymph node development. Remember that this lymphotoxin beta receptor is present in varied anatomic niche, including gut, but its role in those anatomic niche has remained unclear till date. Now, one can easily, conveniently measure the non-canonical pathway activity. If you treat cells with an agonist, lymphotoxin beta receptor agonist antibody, which would activate lymphotoxin beta receptor, it would lead to processing of PIONA-100. You can detect it by immunoblot. You can see a decrease in the PIONA-100 protein level and concomitant increase in the P52. And one can measure the ratio of P52 to PIONA-100 in these cellular extracts as a surrogate of non-canonical NF-kappaB signaling. Obviously, one can also do nuclear NF-kappaB DNA binding assay using EMSA. And as you can see, lymphotoxin beta receptor signaling indeed leads to very weak and sustained activity of NF-kappaB factors. Here you can see two dimers. The bottom one is RELB containing NF-kappaB dimer that I just described. And there is a very weak, if any, relay NF-kappaB activation as well. Nevertheless, the point is that lymphotoxin beta receptor only weakly activates NF-kappaB factors. And this non-canonical NF-kappaB pathway is known to play a role in immune development. It is not thought to be playing a role traditionally in inflammation. However, research from our groups, that is from Alex Hoffman's group, Gauri Ghosh's group, Klaus Saraday's group in MDC Berlin, and many other participants have begun to show that these pathways are interlinked by a varied biochemical mechanism. That allowed us to ask this question: if these pathways are interconnected, would that allow crosstalk between NF-kappaB activating signals? And if so, if such crosstalk would modulate inflammation within the NF-kappaB system, you have an instructive pathway, you have an ancillary pathway, if such crosstalk would modify inflammation. And we started examining it. But we soon realized that examining crosstalk between two pathways could be challenging. For example, when you say two signals crosstalk with each other, would you consider the signals to be concomitant or sequential? If you consider them to be sequential, what are the sequence? Which one comes first? How about the dose of individual signals or their duration? And more so, crosstalk is considered to be an emergent property of interlinked system. So it would generate a nonlinear behavior. It would be also very difficult to intuitively predict if crosstalk effects would be early or late or very late. Further, in a mechanism, in a network where there are multiple such interconnected linkage exists between these pathways, mechanism become very difficult to experimentally address. So one approach that has gained substantial popularity and produced rich dividend with respect to understanding emergent properties of cell signaling network is a combinatorial approach where one can include computational and cell biology, cell biochemistry experiments together, where one can kinetically model, one can reconstruct these pathways, cellular pathways, in a computational model, describe them involving mass action kinetics equation, for example, also describe the interconnectivities in the between these pathways, and simulate thousands of different crosstalk conditions to isolate crosstalk enabling cell signaling regime, and that generates predictable hypotheses. One can then treat cells and test those hypotheses to gain mechanistic understanding about crosstalk, to capture crosstalk for that matters. So we began to thereby start computationally modeling NF-kappaB signaling, a field that is otherwise pioneered by Professor Alex Hoffman at UCLA. But here in my laboratory, we worked with Professor James Gomes at IIT Delhi, reconstructed the canonical and the non-canonical NF-kappaB pathway in silico, describes these molecular species and their reactions that they participate in to transduce signals. We used a series of mass equations adhering to mass action kinetics. And obviously, we had those reactions and rate parameters, around 105 of them. We used, of course, the literature. There is a vast literature for NF-kappaB signaling. We used our own experiment to parameterize the model. And also, of course, fitting exercises were done. In this model, one can measure NF-IKK activity that is induced during canonical signaling, plug it into the model, and simulate NF-kappaB response, real NF-kappaB response. So one can also measure NIC-IKK1 activity induced during non-canonical signaling, plug it into the model, and simulate non-canonical NF-kappaB response. And we did those and then compared simulated response with experimental data for better model fitting. Once model started behaving properly, we started using that model to further understand signaling crosstalk. Now, focusing on we did a lot of work, focusing on the key findings relevant for today's presentation. What we did in one of our analysis, we considered a typical kinase activity profile. What you have is an amplitude. We have durations. A and B together provides for a duration, and H provides for amplitude. So it's a typical kinase activity profile that I'm showing here. What we did next is that we varied these values of H and A plus B to arrive onto a library of theoretical kinase activity profiles. So all possible combinations are there. And then we used our model. We plugged in an iterative model simulation, we plugged in this library through this NEMO-IKK arm, the canonical signaling arm, or through the NIC-IKK arm, the non-canonical signaling, or both. And simulated NF-kappaB responses. We then computed relay NF-kappaB responses in the co-treatment regime relative to the sum of individual cell stimulation regimes to assign crosstalk index. And we asked, what are the kinase profiles that would generate protein crosstalks? Particularly, we focused on the pneumo-IKK kinase activity profiles. We asked, what are the pneumo-IKK kinase activity profiles? What are their attributes that would allow them to engage into the crosstalk of the non-canonical pathway, and that would lead to non-additive NF-kappaB responses downstream? Who are they? So we started pulling out those from our thousands of simulations and plotted them here for their peak duration and peak amplitude values. What we got here is that we found out for varied peak amplitude, pneumo-IKK kinase activity was engaging into the crosstalk with the non-canonical pathway, providing for non-additive NF-kappaB response, synergistic NF-kappaB responses downstream. However, we did not find any single profile of pneumo-IKK activity that had duration of 2 hours or below, suggesting there is a critical threshold of duration. Pneumo-IKK activity obviously can engage into the crosstalk with the non-canonical pathway, but not if their duration is less than 2 hours. And this is when we looked into literature and also performed our own studies. It turns out that there is a disparate kinetic control of pneumo-IKK by inflammatory mediators. For example, cytokines, host-derived cytokines IL-1, and this is what I'm showing you is a kinase assay. IL-1 treated cells, if you examine them, you'll find very short-lived pneumo-IKK activity. On the other hand, LPS treated cells will show you long-lasting IKK activity. So there is a disparate kinetic control of the upstream kinase of the canonical inflammatory pathway. And basing on our model simulation, we argued that IL-1 induced NF-kappaB response would be less impacted if there is a concomitant lymphotoxin beta receptor signaling which induces non-canonical pathway. However, LPS-induced NF-kappaB response will be impacted by concomitant lymphotoxin beta receptor signaling because of crosstalk. We examined those hypotheses in our cell culture experiments in ex vivo settings. These are mouse embryonic fibroblasts. What I'm showing you is that EMSACHR. We found out that IL-1, which produces short-lived pneumo-IKK activity, would produce a transient nuclear NF-kappaB activity. Lymphotoxin beta receptor engagement, as I discussed below, slow, sustained, very weak NF-kappaB response in the nucleus. Co-treatment of cells with IL-1 and lymphotoxin beta receptor agonist didn't discernibly alter nuclear NF-kappaB activities. Now, looking into LPS, which produces a long-lasting pneumo-IKK activity, you could see that there was a little sustained NF-kappaB response in the cell when cells were treated with LPS alone. There was an early peak, and there is a second phase, late phase, which is a gradually diminishing NF-kappaB activity in the nucleus till 24 hours. There's a gradual diminishing activity. However, when cells were treated, co-stimulated using LPS and lymphotoxin beta receptor agonists together, you see this late activity was substantially augmented. This led us to propose that LT beta receptor crosstalk prolongs canonical relay NF-kappaB response to TLR4, but not IL-1R, suggesting that there is a crosstalk, but however, this crosstalk is stimulus-specific in nature, and our gene expression analysis confirmed that many of these genes that are induced by LPS, many of the LPS-induced relay target genes, their late expressions, for example, IL-1 beta, you can see that late expression were augmented when they were co-stimulated through LT beta receptor. So LT beta receptor alone produces a very weak NF-kappaB activity, but when they are acting in a crosstalk settings, they are augmenting, sustaining LPS-induced canonical relay activity and augmenting late expression of LPS-induced relay target genes. And we again combined experiments and computational studies together to understand the mechanism. And this is what we started the lab. We're talking about almost 10 years back. What you see here, the mechanism was quite interesting at that point. We found out, of course, that canonical signaling would activate relay NF-kappaB, which would induce I-kappaB alpha. That's a negative feedback. You will have a dynamically controlled activity of the nuclear activity of relay, which is where there is a clear post-induction attenuation. That is known. But we found out that in our mechanistic investigation that relay NF-kappaB would also induce the expression of NF-kappaB2, thereby would accumulate P100, important mediator of non-canonical signaling. And we found out that concomitant non-canonical signaling would readily convert this P100 into 52, which is known to form ReLB52 dimer. But in our assay, we found out it also supplements to the nuclear relay activity. A mechanism that was not very clear to us then, but we realized that processing of P100 to 52, that canonical signaling would accumulate P100 because of its ability to transcriptionally control the gene encoding P100, that is NF-kappaB2. And that concomitant non-canonical signaling that would convert P100 into 52 would have a serious implication that would supplement to nuclear relay activity, thereby leading to late augmentation of the late canonical response in the TLR region. So at the very heart of it, NF-kappaB2 appears to connect the non-canonical pathway with the canonical pathway and calibrating canonical relay NF-kappaB response. We tested this, obviously, we used NF-kappaB to knock out cells, but we could show that this late effect of lymphotoxin signaling on late NF-kappaB response to LPS was completely gone in the absence of NF-kappaB2 gene. So it suggests that this crosstalk, P100 NF-kappaB2 indeed mediates the crosstalk effect of non-canonical signaling on canonical relay NF-kappaB response. And that is a point where we also begin to ponder over, maybe a bit accidentally, because of one of the students who was very interested in immunology. We were doing immune signaling. My first student, Balaji Benoit. What is the physiological significance of this crosstalk? And that is when we read this paper coming from Yang Zhengfu's lab and Alexei Knumanov's laboratory. They reported that lymphotoxin beta receptor signaling in intestinal epithelial cells orchestrates innate immune response against gut pathogen. So this is what is happening in the intestinal epithelial cells: colonic breach, microbial infection. You have canonical NF-kappaB activation in the intestinal epithelial cells that would promote expression of varied chemokines and cytokines, and that would trigger a cascade of inflammatory events which will contain local infection. Now they're saying that lymphotoxin beta receptor is also important in this gut immune response. We reasoned maybe because of its ability to engage into the crosstalk with the relay pathway, canonical relay pathway via NF-kappaB2. We quickly started testing that. What I'm showing you here is a Citrobacter infection experiment. We infected mice, wild type and global NF-kappaB knockout. That is the point we had global NF-kappaB knockout. We used wild type and global NF-kappaB2 knockout mice, infected them with Citrobacter and collected intestinal epithelial cell and compared nuclear NF-kappaB activation in them. Clearly, Citrobacter infection led to profound nuclear activation of relay NF-kappaB factors. We found out that in NF-kappaB2 knockout mice, epithelial relay activation was weakened. That led to very serious fall, impairment of the expression of Kc and MIP2 alpha. These are important chemokines and cytokines. Impairment of their expression, compromising gut immunity, so as that NF-kappaB2 mice, they succumb to gut infection as compared to wild-type mice where it was self-resolving. So, this is what was the conclusion of that study. Clearly could demonstrate that non-canonical NF-kappaB2 signaling sustains canonical relay NF-kappaB activity, ensuring adequate inflammatory immune response against gut pathogens. To give you the chronology of the event, we used computational biology, computational modeling studies, simulation studies That is instrumental in those early days in my lab to molecularly characterize NF-kappaB crosstalks. But we also took an extra stride and examined the physiological relevance of such crosstalk in animal studies. And that was the beginning of our crosstalk voyage into immunology. And subsequent years, we have published several papers. We have shown that crosstalk is not only stimulus selective, but also cell type selective. We have shown that NF-kappaB crosstalk is an important regulator of lymphocyte trafficking in reactive draining lymph node. We have shown that NF-kappaB crosstalks are important in Treg homeostasis. We have shown that deregulated NF-kappaB crosstalk might be fueling multiple myeloma. And only recently we have also shown that NF-kappaB crosstalk is important for type 1 interferon response. Today, I will discuss some of our recent studies in this subject, where we have focused on immune signaling crosstalk in the context of aberrant intestinal inflammation. And this particular this particular paper was particularly encouraging for us, where trans-ancestral genomoid association study was done, and it was identified lymphotoxin beta receptor and NF-kappaB2 encoding genes were associated with the risk loci of IBD. They are the important mediator of so-called naive, tonic, nimble, non-canonical NF-kappaB pathway. They appears to have an association with human IBD. And what happens in the cholitogenic gut? Because we found that NF-kappaB crosstalks reinforces inflammatory relay response to gut pathogens in the ICs. We initially focused on ICs. In ICs, when there is an intestinal aberration, when it's a cholitogenic gut, you have deregulated canonical NF-kappaB activity that leads to uncontrolled expression of chemokines and cytokines, that leads to pathological tissue distractions, and that perpetuates into colitis. What is not clear is what causes this excessive canonical NF-kappaB activity in the cholitogenic gut. We asked the crosstalk that we discovered that was calibrating relay NF-kappaB response to gut pathogen, would that be responsible also for fueling such uncontrolled inflammation disease? That's the question we set for ourselves. Does non-canonical NF-kappaB signaling modulate relay-driven inflammation in the cholitogenic gut, leveraging this interlinked system? So again, the ancillary pathway, is it now fueling the instructive path in a manner you get disease? We focused on human IBD first. We took around 30 IBD biopsies from 30 IBD patients. They were suffering from ulcerative colitis, and 10 IBD-free control individuals ailing from hemorrhoids. And those tiny biopsies, that's some backbreaking work that Meenakshi Chawla in my laboratory did in the last couple of years. Those backbreaking biochemical analysis done on those biopsies. We examined them for NF-kappaB enzyme. We examined level of protein of I-kappaB alpha and P52, P100. Obviously, those IBD biopsies, we have found out that there is increased relay NF-kappaB activity in the colon, the IBD patients. And this is known. What we further studied is if non-canonical pathway is also activated. And as you can see, we measured P52, level of P52 in relation to P100 as a surrogate of non-canonical NF-kappaB signaling. And what we see here is that there is a frequent engagement of the non-canonical pathway. It's activated indeed in IBD. Not only that, we had another surprise waiting for us. Within the IBD cohort, obviously, they had more NF-kappaB activity in the nucleus, but we found that the patients, those samples, we found there is a distribution. Some patients have very high level of relay activity in the nucleus, some had moderately high relay activity in the nucleus. So we subcataloged those two patients into two subgroups, relay moderate and relay high. And we found out that the relay high subgroup had stronger non-canonical signaling as compared to relay moderate subgroup. So we concluded the strength of non-canonical signaling correlates with the amplitude of the canonical nuclear relay activity. So that was very interesting. Is it, then of course, the next question we asked, is it causal? If non-canonical signaling has a causal role in fueling gut inflammation, to address this question, we resorted to animal studies. We subjected mice to DSS treatment through drinking water. DSS cause erosions in the mucosal layer, cause disruption in the intestinal barrier, and leading to intestinal inflammation. We collected those ICs and examined them biochemically. What we found that in ICs, there is quite a bit of constitutive processing of P100 into 52, far more than what you see in cultured mouse embryonic fibroblasts. However, DSS treatment further augmented P100 processing to 52. So, DSS treatment in mice promotes P100 processing to 52, promotes non-canonical signal. That's the first interesting observation. Then, when you examine those ICs for nuclear relay activity, what we found is this. DSS treatment leads to strong nuclear relay activation in ICs, in wild-type mice. By 12 hours, you see pretty robust nuclear relay activity. Interestingly, this is again a global knockout. Interestingly, NF-kappaB2 mice, knockout mice, which lacks this non-canonical signal transducer NF-kappaB2, which also mediates crosstalk. In NF-kappaB2 knockout mice, we found that there was nuclear relay activation was rather restrained. And with Subra Vishwas from Singapore Immunology Network, we performed global transcriptomic analysis using these ICs. And what we found, and we of course, we ranked first the genes based on the differential DSS-induced expressions between wild-type and knockouts. And we performed gene set enrichment analysis using this ranked list. What we identified here, that there is a significant enrichment of relay targets among genes whose expression, DSS responsive expressions were blunted upon NF-kappaB2 ablation. Moreover, we also did geo-enrichment analysis. Topmost enriched geoterms among genes that were induced by DSS in wild type were related to broadly inflammation and innate immune processes. Interestingly, these geoterms are consistently less enriched in global NF-kappaB2 knockout mice upon DSS treatment. These together lead us to conveniently conclude that NF-kappaB2 signaling augments relevant expression of inflammatory genes in ICs of colitogenic mice. Consistent to, or rather nimble, rather timid inflammatory gene expression in NF-kappaB2 knockout mice upon DSS treatment, we found out that there was reduced recruitment of inflammatory effector cells, for example, Th1 and Th70. DSS treatment leads to accumulation of those inflammatory immune cells in the gut that further fuels intestinal damage, inflammatory damage in the intestine. In the knockout, we see those inflammatory Th1, Th17 cells, their abundance were reduced. And consistently, when we did some of those colon studies, we found out that DSS treatment typically leads to colon shrinkage, but NF-kappaB2 knockout mice, consistent to a lack of, or consistent to a controlled inflammation in the NF-kappaB2 knockout mice upon DSS treatment, they exhibited lesser colon shrinkage. Our HE staining also showed that DSS treatment leads to pervasive colon damage, epithelial damage that was accompanied by a lymphocyte, leukocyte infiltration in the lamina propria. NF-kappaB2 knockout mice, we saw those damages were less obvious, and you can also see some intact villi. We also measured disease activity in these colitogenic mice. DSS treatment leads to body weight loss, leads to stool inconsistency, and leads to rectal bleeding. And you can summarize them in a disease activity index, and you can see that as compared to wild type, NF-kappaB2 knockout mice exhibited less disease upon DSS treatment. So, non-canonical NF-kappaB2 signaling appears to exacerbate indeed experimental colitis. But what is the cell type? We are using global knockout. And by now, we started using, of course, cell type specific knockouts. Here, we are using intestinal epithelial cell specific knockout of NF-kappaB2. We ablated NF-kappaB2's function in intestinal epithelial cells. And that knockout strain phenocopied global knockout. We found out that IC-specific knockout of NF-kappaB2 gene, knockout of NF-kappaB2 led to similarly reduced Relay NF-kappaB activation in the nucleus upon DSS treatment, reduced expression of inflammatory mediators, reduced recruitment of inflammatory effector T cells, and reduced disease activity. At this point, we obviously asked what could be the mechanism? Is it cell intrinsic? To study the mechanism, we started examining this mechanism in mouse embryonic fibroblasts. What we are showing here is that we chronically activated lymphotoxin beta receptor in mouse embryonic fibroblasts for 36 hours, and then treated those cells with LPS to mirror intestinal epithelial niche, where we believe chronic lymphotoxin signaling is impinged upon epithelial cells. And when we did our IMSA analysis, what we found out that as compared to naive cells, lymphotoxin conditioned cells elicited far stronger LPS-induced relay activity in the nucleus. Furthermore, this lymphotoxin conditioning effect is completely gone in NF-kappaB2 knockout mice, suggesting there is a cell-intrinsic crosstalk. There is a cell-intrinsic lymphotoxin beta receptor NF-kappaB2 signaling amplifies LPS-induced canonical Relay NF-kappaB activity. We did far more mechanism studies to summarize what we did observe, and that's a key figure that would come again at the very end of this talk. From studies from Gauri Ghosh's laboratory, Alex Hoffman, TCLA, cross-cytotypes as MDC Berlin, and also our group, we have found out that while P100 plays an important role in non-canonical signaling in regulating nuclear LB activity, it has been shown that P100 and there is another precursor NF-kappaB, P105, they together form a oligomeric complex. Some people call them NF-kappaBism, some call them I-kappaBism. Some groups are have even different names for them. But nevertheless, these are NF-kappaB precursor oligomeric complexes. And it has been proposed that these oligomeric complexes also sequesters relay. And this NF-kappaBism is quite distinct from the latent complex. Latent complexes where relay is held inactive in the cytoplasm by I-kappaB alpha protein. But latent complexes are sensitive, are responsive to canonical signal, where I-kappaB degradation allows for nuclear accumulation of relay. However, these NF-kappaB complexes are not responsive to canonical signal. What you found out, or you can propose that NF-kappaB complexes, this NF-kappaBism, they are sensitive to non-canonical signal. That leads to P100 processing into 52, liberating relay dimer that transits from NF-kappaBism to latent complex. Once non-canonical signal induced processing of P100 to 52, liberating relay dimer that then get readily sequestered by I-kappaB alpha in latent complexes. So there is a movement. Non-canonical signal initiates the movement of relay dimers from NF-kappaBism, which is insensitive to canonical signal, to latent complexes sensitive to canonical signal. Indeed, what you found that very strong non-canonical signal swells this latent compartment, thereby creating a cell state poised for hyperactivation via the canonical pathway. Once the cells experience signals that activate the canonical pathway, you have hyperactive Relay NF-kappaB response in the nucleus, leading to deregulated chemokine cytokine express. That is the very heart of IC deregulation of NF-kappaB, we believe, in the cholitogenic curve. And from there, the conclusion was very simple, that non-canonical NF-kappaB2 signaling appears to, in ICs, appears to amplify canonical NF-kappaB activity, exacerbating intestinal inflammation. And then the last few minutes, I will spend on this, something that we have not even published today. We are almost there. This is about dendritic cells. Non-canonical NF-kappaB signaling has been shown to play an extraordinarily important role in dendritic cells. Work from other laboratories has shown that RELV in dendritic cell has a role in IL-23 expression, has a role in controlling interferon expression, thereby controlling CD8-mediated T-cell response. It has been also suggested that relevant dendritic cell has a role in Treg formation, FOXP3 positive Treg formation. Because there are so many articulation that the non-canonical pathway in dendritic cell plays a role, and dendritic cell, of course, has an important role to play when it comes to intestinal homeostasis and inflammation. We asked if non-canonical NF-kappaB signaling disease also fuel gut inflammation. And to address this question, we first looked into the intestinal disease to ask this question if intestinal inflammation is associated with also an activation of the non-canonical pathway in disease. So this is a data set that we have used from Kasuto Tamai's laboratory. It's a single cell RNA-seq data set, but they, in a time trajectory, performed single cell analysis using DSS-treated mice. We used the data set, particularly that are relevant from mononuclear phagocyte, to segregate macrophages and dendritic cells. And our analysis of those data suggested that dendritic cell express far more relevant NF-kappaB to mRNA as compared to macrophages. Second, in a time trajectory of DSS treatment, when we compared these genes, we found out that non-canonical NF-kappaB signal transducers, relevant NF-kappaB, their mRNA level were increased upon DSS treatment. Not only that, non-canonical NF-kappaB target genes, CXCL12 and 13, we found out they're normally very lowly expressed in intestinal disease, but we found out their accumulation at day 6 in intestinal disease that temporarily coincided with the accumulation of relevant NF-kappaB, the non-canonical NF-kappaB signal transducer encoding mRNAs. So this led us to conclude that non-canonical NF-kappaB signaling is indeed activated in intestinal disease during the progression of colitis. Is it playing a role? What we did, we ablated non-canonical NF-kappaB pathway in disease in CD11C cells. When we disrupted RELV function in CD11C cells, those mice exhibited resistance to acute colitis. They exhibited a readily exhibited resistance to acute colitis induced by DSS. NF-kappaB2, NF-kappaB2, however, when we ablated NF-kappaB2 in CD11C mice, we found out that acute colitis data with respect to acute colitis model, DSS treatment for 7 days, that acute colitis model, when you looked into the body weight change, those mice, they showed only modest difference as compared to wild type. However, when we performed chronic colitis, where mice were exposed to DSS treatment in a cyclic manner, these NF-kappaB2 knockouts also exhibited resistance to colitis. So it suggested that non-canonical NF-kappaB signaling in disease would be fueling experimental colitis. And we looked into the mechanism. First thing we reasoned that in dendritic cell, maybe the non-canonical pathway is similarly fueling canonical activity, NF-kappaB activity, and that is why we see this difference. However, it was wrong. We didn't find out any crosstalk between non-canonical and canonical NF-kappaB signaling in dendritic cells. So you wondered what could be the mechanism. We performed RNA-seq analysis and looked for enrichment of pathways among genes differentially regulated between wild type and NF-kappaB2 knockout disease. And one of the top ranking geoterm was retinol metabolism. So we will look into it. In dendritic cell, retinol is converted into retinoic acid with this family of enzyme called RDHs and RALDHs. And dendritic cell produced retinoic acid, particularly in the gut, has a very important role to play. It supports the sustenance of FOXP3 positive CD4 cells in the gut. It also promotes IgA positive plasma cells. Thereby, it promotes luminal IgA level, which controls microbiome. We looked into those enzymes belonging to the RDH and RALDH family. And we found out very interestingly, the deficiency of the non-canonical pathway, be it in the form of RALB, be it in the form of NF-kappaB2, leads to heightened expression of RALDH2 mRNA in BMDCs. Not only expression of RALDH2 mRNA was high, when we did an activity assay, we found out that deficiency of the non-canonical NF-kappaB signaling leads to heightened RALDH2 activity. Obviously, we looked into the intestinal disease then. As such, deficiency of non-canonical NF-kappaB signaling in these knockouts did not alter the frequency of dendritic cell in the intestine, neither did it have an impact on the expression of CD8 T6 and CD8 T. Not a significant impact we could observe. However, deficiency of RALB resulted in increased RALDH2 activity in intestinal disease. So indeed, even in the intestine, deficiency of the non-canonical pathway promoted RALDH2 activity, which is known to play a tolerogenic role by expressing retinoic acid. Was that consequential for intestinal Treg population? We collected intestinal lamina propria and mesenteric lymph node cells, and we found out that absence of RALB in the CD11C cells led to profound accumulation of Tregs, FOXP3 positive T cells, in both lamina propria and in mesenteric lymph node, both in untreated as well as DSS treated RALB knockout mice. Similar results were obtained also in NF-kappaB2 deficient mice, suggesting that this pathway in dendritic cell actually limits accumulation of inflammation suppressive FOXP3 Treg cells. And mind that we looked into the IL-10 expression of these FOXP3 positive CD4 T cells, and they were equivalently expressing IL-10 as much as one can see from wild-type mice. And this work was done with Amit Awasthi at THST in collaboration. We asked if these dendritic cells are showing heightened RALDH2 activity, did that impact IgA positive BT2 cells? Indeed, it did. We found out that a deficiency of RALB in CD11C led to increased IgA positive cells in the lamina propria, and that resulted in increased luminal IgA. And this led to changes in the microbiome, or microbial, we sequenced the gut microbiome, compared wild type and knockouts. And this was done in Bhabhat Das, is the local institute THSTI. Indeed, we see changes in the gut microbiome. This was very instructive changes, particularly if you look at the phyla, you see a decrease in the actinobacteriota and increase in firmicutes. Many members of firmicutes are known to play an important inflammation suppressive role. Particularly when you look into the microbial genera, we found out lactobacillus. That level, there was an increase in the lactobacillus level, which belongs to this phyla firmicutes. And we are further characterizing it, but the message is very clear that deficiency of non-canonical NF-kappaB signaling in dendritic cells leads to heightened RALDH2 expression, and that produces a tolerogenic gut by instructing Treg and Ig axis. We asked what augmented RALDH2 expression in this knockout disease. RALDH2 in disease are expressed involving 3 transcription factors, NOTCH2, beta-catenin, and IRF4, and these transcription factors are regulated at the level of protein expression. We looked into those proteins and looked for their expression level and found out indeed absence of non-canonical pathway in BMDCs led to increased beta-catenin level, and our inhibitor study suggested that beta-catenin was important, at least in BMDCs, in promoting expression of RALDH2. We immediately looked into intestinal disease again. Deficiency of the non-canonical pathway led to increased beta-catenin even in the intestinal disease. And this is a paper that was very interesting, and that gave us a lot of impetus. Is from Balipulendran's lab. This is a bit old paper, which suggests that the beta-catenin in dendritic cell indeed plays a role in tolerization of gut. So looked into the beta-catenin pathway very carefully. In resting cell, beta-catenin is degraded by this destruction complex consisting of APC, GSK3 beta, and CK2. Auxin family of protein tethers beta-catenin to the destruction complex, leading to its constitutive degradation, whereas Wnt rescues beta-catenin from this constitutive degradation. Our analysis did not support a role of cell extrinsic Wnt ligands in our phenotype. We then looked into intracellular regulators of Wnt, and we got extremely excited because we found out that auxin-1 mRNA levels were decreased in RELB knockout BMDCs. And in partnership with Professor Alex Hoffman, UCLA, who could demonstrate that RELB indeed directly binds to auxin loci. It binds to its promoter, and there is this intronic enhancer where also RELB binds. Indeed, this appears to be the RELB directly controls auxin-1 transcription, and absence of RELB in BMDC led to reduced auxin protein, and our immunoprecipitation reaction suggested that led to decreased binding of beta-catenin to the destruction complex, leading to its stabilization in the absence of non-canonical signaling. Currently, we are, of course, looking to the genetic model. What we are planning to do in this last experiment, we are hoping to finish by this month, and we will submit this paper. If beta-catenin haploinsufficiency in the background of non-canonical NF-kappaB deficient tissue would diminish RALDH2 expression and would reinstate cholitogenic sensitivity. While we are doing this experiment, there's a recent paper published in 2020 shows that lymphotoxin beta receptor indeed could modulate Wnt-induced pathways in lung. Enthused with those circumstantial evidences, we nonetheless proposed a model of crosstalk between non-canonical signaling and beta-catenin pathway in dendritic cell. In this mechanism, RELB promotes auxin-1 expression, and auxin-1 promotes degradation of beta-catenin. When there is a controlled non-canonical NF-kappaB activity, you would have some beta-catenin that would go into the nucleus, promotes expression of RALDH2, and instruct gut homeostasis through Tregs and IgA microbiome axis. But strengthened non-canonical signaling would lead to pervasive degradation of beta-catenin, weakening this Treg and IgA microbiome mediated gut control by beta-catenin. And that is the tolerogenic gut. So we conclude that non-canonical NF-kappaB, RELB NF-kappaB signaling in DC restrains the tolerogenic beta-catenin RALDH2 retinoic acid axis to promote intestinal inflammation. To sum up, we have shown you that this pathway is crosstalk. At the heart of it is this non-canonical pathway, which crosstalks with the canonical pathway involving this RELA NF-kappaB axis to modulate inflammatory activity in the intestinal epithelial cell. We have also shown that non-canonical NF-kappaB pathway crosstalk with the beta-catenin axis through RELB auxin-1, RELB auxin-1 involving RELB and auxin-1 mechanism. And taken together, this crosstalk promotes gut inflammation. Take-home message: we have established that pathways crosstalk, and crosstalk between immune signaling pathways could be probably a dominant immunoregulatory mechanism when it comes to inflammation control. There is a hope also that you have these central pathways that transmit signals, and for example, an instructive pathway, we cannot target them therapeutically, fully knowing that is the one that is instructing inflammation, because it would cause devastating side effects. We propose that our study gives a hope that one can instead target this cross-regulatory mechanism, thereby achieving disease-specific intervention, cell type-specific intervention. So here I will probably conclude, and I will again thank for the patience. And this is what we do. We study this complex immune signaling mechanism to further our understanding of human health and ailment. Thank you very much for your patience. And it was wonderful to share some of our published and unpublished data. Thank you again.
Well, thank you very much, Soumen, for sharing this great presentation with us. I'm sure that there's many questions, but as you know, as usual, you have to ask those questions with a webpage. I'm just waiting actually now to see the webpage information here. Here it is. Yeah. So please ask many questions on this platform formerly known as Twitter, and we look for intense and challenging discussions. And with that, I'd like to thank you very much again, and I'd like to close this session now, or this seminar. And I wish you all a great remaining day, and I hope to see you again next week when our speaker will be Amanda Lund. Thank you very much, and see you. Bye.
Thank you.